I would point out the first book I ever worked on was this thing. It was called Some Mathematical Questions in Plant Biology. It was part of a lecture series on math and the life sciences. And this was done, it's hard to believe, 30 years ago. And so this was a collection of talks, including one by Carl Nicholas, who you may have read. He's done an awful lot since on plant allometry and evolution of plant form and structure. Richard Rand on dynamics of stomata. John Thornley and Ian Johnson. John, if you've never heard of John Thornley, he was one of my major mentors in terms of applications of mathematical approaches in plant science. And he and Ian Johnson had an article on crop growth issues in this. And Hal Caswell, who you probably know from the Bible of matrix population modeling, did a lot of work on plant demography. And the other author in this was Joan, when Joan was John, Roughgarden, on theoretical ecology of plants. And so there wasn't any disease stuff in this, okay? I think if we were to redo it now, there would definitely be a sort of whole series of chapters, perhaps written by many of you, on issues of plant disease. So that's a nice little book. And then the most recent one, this is what happens when you age, see? And you get bigger. So Alan is downstairs, and several of you, including Sherry, did an article in this. And who else? Kevin? Kevin did one. Did anyone else? No? OK. Oh, Linda did that one. That's right. Yeah. So if you're interested in taking a look at it, I don't have them for sale, but encourage you to take a look. There's lots of sort of background articles in there on many areas of application that we encourage authors to write specifically for graduate students to provide an intro to different areas, whether it was host parasitoid or whether it was spatial modeling, whatever it was. So my challenge today, I understand that because we have a mixture of people here, I was going to talk about a sort of a little bit of background specifically in modeling issues. But I know that Mike is going to talk about transmission and models too, so there's going to be a lot more detail. So this is going to be a little bit of a broad brush, and hopefully you won't be totally bored, okay? So we'll see. So I wanted to start out with a case study on disease mapping that grew out of a workshop here, and then some comments on modeling and models, and they're not the same thing. And then there was a paper. How many of you have ever heard of the journal New Phytologist? Oh, good, a large number. OK, that's great. So there's a paper, actually a letter on 100 plant science questions, and I'll focus on the disease part of that.
On the WordPress site.
Yeah, there's a link to it on the WordPress site, hopefully. And then I'll go on and just end up with some example papers. When I was asked to do this, I went and just sort of looked for the last decade of things and pulled out a few examples, okay, which some of you have worked on. So we had a workshop here on mapping of infectious disease, and that led to this paper in Transactions B, the Royal Society. And it came out in 2013. The workshop was about 2 years before that, all right? And I think it's of particular interest here because, let's see, I'll put this up. This is the overall, this is the abstract of it. So don't worry about it. It's like, you're not going to read it, OK? But it was to look at the geographic distribution of infectious diseases, all infectious diseases of clinical significance to humans, OK? And so there were 355 infectious diseases looked at in this paper. And they found that in this case, about half of them had a strong rationale for looking at what the global distribution is and formally mapping them. But only about 7 had been comprehensively mapped. So that was part of the objective of this workshop, was to sort of start this process of looking at disease maps. And just to give you some idea, this is one page from the supplement. So there's 355 pages, okay, of supplementary material that include maps. And I'm going to blow this up because you can't read that. So the upper part of it has a map. It talks. In this case, I chose a vector, a tick-borne disease. So figure that you might be more interested. It's not plants, but at least it's ticks. Anaplasma. And so it starts out with a little bit of the taxonomy. So in this case, it's a rickettsial. And a little bit about the natural history, and a bit about the fact that, what are the estimated R0 values, the rate at which secondary infections arise from a primary single infection in a completely susceptible population. In this case, it's not humans, but in the reservoir populations, there's a range of possible R0 values. So this is sort of one part of it. The other part of it, so this is the second half of that page, and it basically talks about how much interest there is in terms of hits on the databases. It looks at where it's endemic, how many countries, and where the sort of previously published maps were. It gives references to them. And the important thing that I think we really encouraged, and what happened, what grew out of this workshop, was some idea of the data quality associated with this, okay? And then a recommendation on how to proceed with mapping, all right? Now, does anyone know, is there anything like this for any plant disease? I looked. I couldn't find, any kind of compendium. I know there's ones for individual diseases, I think, but
there are distribution maps published by CABI across a whole range of different organisms, fungi, bacteria, and so forth. I wouldn't make any comment on usefulness of them.
Okay.
But they certainly exist. And there's the CABI compendium as well, which to some extent does this on a just as this does on an individual disease basis,
okay.
pathogen basis.
Okay. I couldn't.
Not to the level of analysis that's gone into that.
Okay. So this is data-driven completely. There's not much in the way of models underlying any of this. Okay. But as you all know, data is really important to drive models. So. I thought I would just point that out as one thing that came out of a previous workshop here that you might think of as a possible component of something in the future. So going on to talking in general about mathematics. So for the people that have taught mathematical modeling courses, you can go to sleep or you can check email, whatever. But I do want to give a bit of an overview. There are alternative ways of expressing theory, and so I put them down here as verbally, graphically, mathematically, and through simulation. And there are a variety of approaches to develop theory. So classically, there's empirical and or statistical approaches, and there are comparative approaches. So there's a whole society of integrative and comparative biology, among other things. And then there are approaches that are more mechanistic, in which you try to come up with an understanding what the underlying processes are. Often those are done by breaking things up into compartments, and many of the models in disease ecology are broken up by compartments of susceptible, infective, remove kinds of things. And then there are optimization approaches in which you look at sort of what's the best that could occur according to a range of criteria, and can we get there evolutionarily, right? The adaptationist view. Then there are systems approaches, and now there's this whole field of systems bio. We're not going to try and define it here, but there are hierarchical approaches. There are individual-based ones or agent-based ones that actually grew out, and at least in ecology of another book and thing that happened here on this campus. And there are expert system approaches. And you could add to this network approaches. There's probably a bunch of things that could be called statistical approaches in there that can develop general theory. If you haven't seen it, there's a nice book on the role of theory in advancing 21st century biology that the National Academy did. It's available for free at nap.edu. And it provides a nice overview. If you look at it, though, there's virtually nothing about plant disease stuff. I did a quick look through it. It's not much there. Okay, but That's okay. I mean, there's many dissertations to be done, okay? And sort of there's a general taxonomy of different kinds of models. And I already sort of mentioned one. So one taxonomy is conceptual. And often when we start out with a modeling process, you do some kind of a conceptual model that's a kind of graph of this interacting with this, interacting with this. They can be verbal models as well. They can be explicitly quantitative. And physical models are ones in which you're actually using an organism. So when I talk to many biologists about models, the first thing that they think about are rats and mice. Okay, they're not, a model is a mathematical model. So I've found to be really careful when I talk to a non-modeling audience about what a model is. Okay, so they're typically physical or biological models. And often modeling textbooks sort of classify things based upon the mathematical approach. Now, I've put together a list of what I think are reasonable objectives for models. And there's lots of different objectives. This is page 1. And so suggesting observations and experiments, providing a way to assemble bodies of information and facts. This is one of my favorite from François Jacob. Allows us to imagine and explore a wider range of worlds than ours, giving new perceptions and questions about how our world came to be as it is. So exploring alternative universes in some sense. It also can clarify hypotheses, develop key components and systems, and it allows us to investigate situations that we might not be able to otherwise investigate because of constraints, societal constraints or ethical constraints and so on. So, you may not want to carry out a certain vaccination scheme on humans, but you can play with it in the model and not worry about it. It's only bits. So another range of objectives as models are, it allows simultaneous consideration of spatial and temporal things. So that's difficult often for people to understand. Extrapolating to broad spatial or long temporal scales. It prompts testable hypotheses, serves as a guide to decision making. I like this one. Provides an antidote to the helpless feeling that the world is too complex to understand in any generality. It provides a means to get at general patterns. Okay? But often when people think about objectives of models, they're often after prediction. Okay? And I just wanted to point out that there's lots of reasons to do models, and prediction is only one of them. All right? And it helps to think before you sit down to do any of this why you're doing what you're doing. And it might be several of these objectives. There's no reason to not have many of them in there. Okay? But no one model can do everything. So Richard Levins, in a classic book on evolution and changing environments, at the very beginning of the book has just a paragraph or two that talks about the fact that no one model can do everything. And this is my way of sort of visualizing the trade-offs that he mentions there between generality, meaning a model that is broadly applicable to many systems, realism, meaning that you can actually get the parameters and fit them to data and actually relate the variables in the model to observations, and precision, meaning that these are exactly able to be fit by data sets, okay? And produce results that are precise. And so much of the modeling in what I'll call disease ecology fits over here. They're often models for general theory development, and they're not necessarily very realistic. Down in there are more system simulation kinds of things, nor are they necessarily very precise, which are often statistical models that are fit to a variety of data sets. And I think it helps before you sit down and think about any particular model where your objective is in terms of where it fits in this space of trade-offs. Okay? And there are other trade-offs, accuracy. There's all kinds of other rubrics for doing this. This just happens to be mine based on Levins. Okay. And the other thing is that this is from our Atlas project, just to point out that modeling is an iterative process. It involves a variety of components that starts with data and moves on to sort of deciding upon what the key components are, and then writing down some kind of equations for them and simulating it and evaluating it and analyzing it, and then potentially providing management input, and then producing more data for monitoring and continuing. So the great thing about this is that as a modeler, your work is never done. You keep on going around this, and you always got a permanent job. And you always improve in some sense. But from the perspective of management, at some point you have to say, stop, this is good enough for us to be able to say that this is useful in terms of whatever practical thing. Now, this is designed for environmental systems in which you actually do have a management outcome. There's lots of other objectives for models. You may not want to do that. Okay? But, and there are constraints. Constraints on data, that you may not have the available data to do what you want. There may be effort constraints, how much time you have to put into it. There may be computational constraints, even today with our massive supercomputers around. And there can be other constraints as well. And it's helpful to think about those constraints in terms of what you include in the models. Sometimes for large projects, I'll use the Everglades one as an example. When Don DeAngelis and I sat down to map out what we could do to look at the impacts of alternative ways of managing water flow across South Florida on the biota. And we were just generally charged with all the biota. And of course, you sit down and you think, oh, wow, there's all these great fish. Let's think about all that. And eventually you get down to, we had like a list of like 150 species. And eventually we said, this is ridiculous. We just can't get the data on them. So we wound up with big fish and small fish. Basically that. I mean, piscivores and planktivores. So it's important to think about the data constraints on what you have and effort as well. So in this, the whole process of modeling is a process that I've called selective ignorance. You selectively decide what to include and what to ignore in your model because you're not going to include everything. Otherwise, you'd have the real world. Okay? And just as public policy decisions involve a balancing between what various stakeholders might want, the process of modeling also involves, sort of different approaches and looking at trade-offs in terms of what you include and what you do not include. So I call realistic modeling the science of the actual rather than the science of the ideal. Right? So often in applications, we're interested in getting to the point of being something that a public policy decision would take into account, or a manager or a farmer in this case. Now, there's lots of different terminology associated with how you assess whether or not a model is any good. Right? And this is my way of writing down what some of those terms mean. And there is disagreement, particularly among different fields, on things like validation. All right? So I've said here, validation is a model is an agreement with the real system it represents with respect to the specific purposes for which it was constructed. So, well, that has underlying it a couple of things. One is some notion of accuracy, okay, because if it's an agreement, how far off is okay, right? And it has to do with the fact that there's some domain of applicability. So there's some constrained region, okay, in which you're going to be able to say that this model is useful. And so one of the National Research Council committees I was on models in the environmental regulatory process for the Environmental Protection Agency, which uses models all the time for a variety of things. And it took us 3 years. And one of the things that this group of people that involved modelers, environmental tox people, and chemical folks was that we decided we'd never use the term validation in the book, okay? Because there are really different communities that have very different views of what that term means. And if you're coming out of a statistics background, it means something very different from when you're coming out of an engineering background, okay? So I just encourage you to be careful with that. Some of the other ones, verification typically means that, yeah, I mean, the model's doing what I constructed it to do in terms of the code is verified and so on. And calibration typically means that there's some data set that you didn't use in developing the model that you're using it to compare to. And so sometimes it's called tuning, and corroboration is similar. I try to use the term evaluation or model testing for the general process of assessing whether or not a model is useful for the purposes for which it's been constructed. So given that there are many different objectives for models, you should think also about many different ways of evaluating those models, that there might be different criteria for assessing and evaluating whether a model is useful. And I really encourage you to think about building those criteria in before you invest time in the model. I've tried to do this with our students in ecology. I think I get further along with them than I do with the math students, who basically want to sit down and write down a set of equations and don't even want to think about data generally. Okay. Now it's not all of our students. Okay. But okay, there are exceptions in the room. So in doing this, you should probably think about data constraints and other things too. So and all of this comes into deciding what it is that you're going to ignore and what you're going to include too. So this is some comments on evaluating different types of models. So in that triangle I put down, there are models for theory development, and for those models that are general and have not designed to be precise. Then what you're often after are qualitative ways of comparing the results of the model to patterns that are observable. Okay? In fact, there's a whole approach to modeling called pattern-oriented modeling that grew out of the agent-based folks. So Volker Grimm, Don DeAngelis, Steve Railsback, a whole bunch of other people have published a couple papers in Science on pattern-oriented modeling. Okay? The objective is to get at understanding how a variety of patterns arise and coming up with alternative models that might lead to those patterns. So in general, you're not doing calibration or corroboration there. Okay? But whereas for descriptive models, then there's this whole field of model comparison and statistics. Tremendous number of books written on this that are designed to sort of help you choose models, information criteria, and others. And then when you have models for specific systems, you're often there involved by quantitative comparisons. And in this case, comparing things component by component, all right? And maybe subcomponent by subcomponent. So that's just a general thing. I've tried to take this into account when I get papers to review. These are the kinds of questions I ask. And often it's for a biology journal. Okay? And so are the models appropriate to the biological questions being addressed? Does it make sense to use this set of differential equations, or does it really need something else? Are the underlying biological questions of potential interest? In other words, is the biology interesting, or is this just a made-up question that somebody's thrown in there? Does the mathematics teach us anything new that's biologically significant? It helps if the mathematics is correct. If the paper is strictly theoretical, does it point out to broadly useful new insights? And are the model parameters and variables estimable from observations if it's indeed not after general theory? So those are the kinds of questions that I ask myself when I get a paper to review. And I encourage you to think about that. So some take-home lessons on model evaluation. For all types of biological models, I think you'll find that evaluation is pretty rare. So I published a paper in a journal called Biological Theory sometime in the last year, in which I looked at a couple of journals. I looked at a series of AMNAT journals and a series of, I can't remember, American Naturalist, and might have been, was it Evolution? No, may have been Ecology. And I looked for all the papers that dealt with models in it, and I looked to see if they ever had any mention of evaluation of the model. And it was extremely rare, extremely rare. And then setting criteria for evaluation prior, tying the evaluation criteria to the model objectives. We're not going to talk, I don't think we have a session on education, but I always try to encourage people to think about educational initiatives. And we have a whole evaluation process here for every activity at NIMBioS. And I encourage you to think about evaluation associated with all your teaching that you do, if you're doing teaching too. Multiple models are good. I encourage you to think about multiple ways of thinking about any particular problem, and consider whether an evaluation has been done whenever you review a paper. So now, I want to go on next to this issue of this. This was a paper that appeared in 2011 in New Phytologist. It really was under the letter portion of it. And it was on 100 important questions facing plant science research. The authors for this are here. And I don't think anybody in the room nobody in the room was an author, right, of this thing. No, OK. But it's heavily UK oriented, OK? You probably know many of the people that are on this list. But so what was the procedure? So they posted basically an open set. Please send us what you think are the most important questions in plant science. And that generated a list of 350 questions. They then had a workshop. About 15 people attended this workshop. They spent a couple days compiling those 350, putting them in groups, and deciding amongst themselves, these are the most important ones. Okay? And that's sort of what they led to. So I found it interesting to look at this paper because although I've done some plant stuff, I'm mainly doing vegetation and other things now. So what they did is they grouped the questions based upon issues of societal questions, environmental and adaptation things, species interactions, understanding, utilizing plant cells, and diversity. And that was basically the grouping of the questions that came in. And just to give you an example of all the top 10, so under society, these were the 4, and then there's another slide with more of them. How do we feed our children's children? Which crops can be grown? Where? What are the best ways to control invasives? So they're fairly broad, general questions that you could spend your entire life, devoted to addressing. That's the first one, and here's the other ones from that list. How do we ensure that sound science informs public policy decisions? Well, that's not something that's just limited to plant science research. That's an issue on all the things that we do. But what new scientific approaches will be central to plant biology in the 20th century? You could take out plant biology and put on any field, whatever, there. So they're kind of general questions. But there's much more specific ones when you go to this. So under species interactions, which is where you would think most host pathogen and disease things would wind up, and that's where they are in this. I'll highlight these, OK? What are the best ways to control invasive species, including plants, pests, and pathogens? How can we provide a solution to intractable plant pest problems in order to meet increasingly stringent pesticide restrictions? Pesticide could put in herbicide or anything there. Is it desirable to eliminate all pests and diseases in cultivated plants? I'd actually never thought about that. Is it desirable to get rid of everything? I hadn't thought about that. But what's the most sustainable way to control weeds? So this is the first part of this series. And there's other questions there, but I'm highlighting the ones that I think are of potential particular interest to this workshop. And then here's another set. How can we use our knowledge of the molecular biology of disease resistance to develop novel approaches to disease control? Okay. What are the mechanisms for systemic acquired resistance to pathogens? When a plant resists a pathogen, what stops the pathogen from growing? How do pathogens overcome plant disease resistance? And is it inevitable? That's an interesting question. Can we use non-host resistance to deliver more durable resistance in plants? Now, I think probably every one of these questions has a theoretical component. Some of them are evolutionarily based. Some of them are more mechanistic. And probably we could structure different breakout groups around these kinds of questions. Some of them overlap, definitely, although there wasn't one about animal-plant, sorts of things there. So I encourage you to look at this paper, and it provides a nice kind of background, at least of what some people think of as important questions in plant science research that directly relate to this. Now, what I then did, Mike, I figured I'd start with this. what I then did is I said, all right, let me look at what kinds of papers are published that deal, and I did a Google search specifically on math models and vector plant disease, virus, I think I said, virus too. And not an awful lot came up, okay? You know what came up most? This workshop. No. Yeah, but I wanted to do, and so Mike is here and he's going to talk about this. But I wanted to choose a set of papers to talk very briefly about the kinds of mathematics that has come up in these, and this one comes up. So Frank can't be here, and I guess Holt. I don't know, Madden. I guess they couldn't be here either. So. We have one representative, but the point here is that often these are mixtures of dynamical system models, typically differential equations. I actually tried to find some discrete time models, which I thought would be more appropriate, but I couldn't find them anywhere. There must be one example. OK, great. So they're often basically systems of differential equations in which you're keeping track of whether or not a host is infected or not infected, and whether or not a vector is infected or not infected. Okay, and then Mike will tell us a lot more about this tomorrow, right? Okay, so but this is sort of one form of it, and this one was 2004, and there's loads of other ones, so I just chose this one, but. And then there are what I'll call them simplifications of this approach. This is a paper that Luis Gordillo, who's at Utah State, I think, is that where he is now? And a collaborator did. In this case, looking at saying, let's assume that the population size is constant, and we can actually reduce the equations then, and essentially turn it into a system of two equations and do sort of stability analysis and. He couldn't come. Yeah, so I figured that everybody, so this was a sort of another one. And this, what's interesting to me, by the way, that I think is a major change since we did this book 30 years ago, and that is that the papers that you're seeing here are published in biojournals. They're not just in math journals. Okay. So that means that there's been this transition where they're not thinking of this weird math stuff as being not related to the biology. Okay. Well, I don't know that I had success, but the field has changed. Let's put it that way. The field is, yeah. Okay. And so here's another one. And in this case, I put this up. This is Rusi, and this one came out just last year in environmental entomology. So one of the this is. No one here is a co-author. Is there?
Well, Sanford.
Oh, Sanford. Couldn't come. Yeah, OK. And Richard Gomokaiewicz has been here many times. So in this case, the reason I chose it is because it's an attempt to look at host plant choice, OK? Because there's good literature that talks about the fact that vectors choose certain plants. And in this case, the paper is looking at the infection state of the plant and whether that changes whether or not a vector might choose that plant or not, and provides the feedback and the sort of the dynamical analysis of this in a general way. By the way, this talk, it will be posted on the whatever site. We'll throw it on the. Yeah, all this is. It'll be the PowerPoint will be on the. I just haven't posted it on there. Yeah. So that's another, but it's still an example of this way of looking at breaking things down into infectious or susceptible or and so on. Okay. And so this is another one. This Holt's on this one too, and Zhang I don't know, but so this came out in Phytopathology. It's again a sort of four differential equation kind of model. But in this case, it was looking at a mild strain and a more virulent strain. Okay, so it's looking at the potential for what's called cross protection, which the formal definition, I guess, somebody can give better than I could, but if you're infected with a mild strain, you don't get the resistant strain or the virulent strain as much. Yeah. Okay. So again, it's a dynamical systems approach, a typical differential equation approach to looking at the general patterns that arise and what will happen there. Okay. Then I found at least one paper that is from Mark Chaplin. I think Ananthi, does anybody know Ananthi? I think he's a graduate student. He may have finished, but it's possible. At any rate, I think this is his dissertation work in which he's trying to construct an individual-based model, okay, of aphid, parasitoids, plants, and virus. Okay, so that's pretty complicated, all right? And he's motivating the movement patterns based upon a standard reaction diffusion kind of. This is straight diffusion kind of model, and this was the only paper I could find published on this. So, but maybe there's something more recently. But the point is that there's a agent-based. The point is that this is a different approach. It's an agent-based approach in which you're looking at movements, in this case, driven by some underlying compartment model or diffusion model. Okay, and then there was this one, which I looked at economic. I tried to find some stuff on economic. I just wasn't looking in the right place, but there's this one was looking at alternative disease control methods for plant diseases in which there's an economic threshold in which you change practice once you've hit a threshold. Okay, they're smiling here like, oh yeah, we know all about this. Okay, and this is Robert. Do you all know Robert? Yeah, Robert. He added the question mark to his name so that when you Google search, you'd actually get him. Okay, and he has a nice little book on epidemiological model, and he's famous because of zombies. Yeah, because he did the zombie science, zombie, and he got to be there with George Romero and all these other famous Night of the Living Dead folks and be on TV and all. It's great. But it was a whole group of students involved in writing that zombie paper, which I thought that was wonderful. Okay.
I was there.
Oh, you were there?
Yeah, it was a course project. Students brought up the zombies idea themselves.
That's great.
Yeah, because he gave a list of the topics. Then students said, I saw the zombies interesting, somehow like a disease transmission as well. And Robert promoted this idea in that kind of. So
that's great.
People criticize the scientists use the money to do zombies. Oh, that's wrong. It's the students' idea.
So are you a co-author on that first zombie paper?
I'm not. I was just sitting because I was postdoc working.
Oh, I see.
Is that the reason why you moved to Beaver's Beaver?
Okay. Well, at any rate, the point is that I tried to find some things that really related to economics, and I probably was not looking in the right literature. I'm sure there's a whole bunch of management stuff there, but I didn't see it in my quick purview. Okay. And then there's a set of issues of sort of within host competition between viral strains, and there's a whole literature on competition models, and within the immunology field, so HIV, and there's loads and loads of papers on within host competition of strains, and I think this paper, I don't know these folks at all. They're at Montfavet, and I don't even. Yeah, do you know these people? Yeah. Okay. So there's a lot more in the paper than just this. I'm just pulling out sort of one sort of. The idea is to look at competition between viral strains and then use sequencing analysis. It's a very detailed paper that I didn't try to go through the details of, but they use stochastic sampling and alternative things in there. Here's another one sort of within host. So this was a cell-to-cell movement of viral strains. In this case, it's a stochastic model for the likelihood of one strain moving between cells, and then looking at the genomics of it. All right. And this one, when did this come out? 2010, I guess. Okay. And there may be a whole literature on this that I didn't find, but it's it was one of the few really stochastic models that I could find. In this case, at a scale that's very different from the scale of the others. Okay. This is potato, isn't it? Oh no, wheat. Wheat mosaic. Wheat mosaic. Yeah. Yeah. This is in wheat cells. Okay. I'm sure there's a bunch of simulation models out there. This was one that I just sort of found, and again, it's somebody that I don't know at all. But I couldn't really write down any equations because it's a simulation model. So, but it's a spatial simulation model. There's probably bunches of those around too. I remember some from years ago on, one of the Cornell folks. Was it Bill Fry, maybe, who did that? Yeah, I don't know. Okay. Yeah, way back. Yeah, and there was actually a code that they released. I think there was a. Yeah. Okay, so what's missing from this stuff, right? So I didn't include it. I didn't find. A quick perusal. I didn't find papers that used network approaches. So whole broad new field of network connections. You're going to say something about it? Are there? Oh, well, I mean, I didn't find it in. You are surprised, or you aren't surprised?
No, I'm surprised.
Oh, okay. Maybe I wasn't looking in the right place, Mike. I didn't see much, but I'm sure it's out there on spatial stochastic models. There's probably a whole field of stochastic models, spatial statistical models that I didn't run across in there. There may be other things. So I'm at the end of what I wanted to say, and I think I'm on time. Am I right? I hope. I'm a little over time. Oh, crud. All right. Okay. Do you want questions, or you do? Okay. Yeah, sure. And I'd be very interested in hearing comments about, oh, you left out all this stuff. Okay. Or whatever.
No, I was given.
Okay.
Well, thank you.
Okay.